So in addition to transmembrane proteins being helical proteins, transmembrane proteins can also, or integral membrane proteins, I'm sorry, be sheet-based proteins. So as an example, we've got maltopore in here, which is multiple, sorry, MULT, multiple multiple polypeptides that assemble as beta barrels, and between them they're able to open up a pore. So that's one way in which sheet protein can assemble and basically enable something to move across a membrane. So in the next chapter we're going to be talking about we're going to be talking about membrane transport and movement of molecules across a membrane, but integral membrane proteins do not have to be alpha helices. They can be beta sheets. And, you know, there's this other instance of the alpha hemolysin toxin. What that looks like is that, our side view is pretty crazy and cool and complicated, but then our top-down view, it almost looks like a straw or something, basically, where metabolites can flow through there and everything like that. Now, as we looked previously, there are lipid-linked proteins, which, there are lipid-linked proteins where a protein has a post-translational modification of a lipid molecule that connects it to, that basically gets inserted into a membrane and enables it to be associated with that membrane. So some proteins are associated with membranes through covalent attachment to lipids that anchor those proteins. Now, one example of that is the addition of these groups right here. This is a farnesyl group. This is a geranyl group. Okay, so this is 20 carbons. This is 15 carbons. Now, these molecules, these groups are these are isoprenoids. But ultimately what you have there is you have these attachments that these are very, very hydrophobic, in which case when they get inserted into a lipid bilayer, they're going to be just happy as a clam because they're associated with these highly hydrophobic groups within that lipid bilayer. Okay, so another way that a protein can be associated with a membrane is going back to our discussion of different phospholipids. So we could have a protein that is right here attached to a phosphoethanolamine head group. Then through several sugars, you have a phosphatidylinositol group that is integrated within the membrane. So we already talked about glycerophospholipids, but this is an example of a glycerophospholipid where our head group, phosphatidylinositol, has some sugar linkages that are then phosphorylated that give a connection to your protein itself. Now, last thing that I want to talk about with respect to lipid-linked proteins is the 4 different types of lipid-linked proteins. So one of the things that we need to kind of draw our attention to is, again, inside of the membrane, outside of the membrane. Okay.
Outside the membrane.
So our 4 different types. We've got a thioester-linked acyl anchor. Can you say thioester?
Thioester anchor.
Yep, a thioester-linked fatty acyl anchor, where we have a palmitic acid group or a palmitoyl group that has been added to a specific either cysteine or a serine residue. Okay, so now another this is a reversible linkage. So this is something that a protein can have this thioester linkage and be connected to a protein. Oh great, Walter's awake. My other son is awake. So you can have that. In addition to that, you can have an amide-linked myristyl group. So a myristic acid is attached to the N-terminal glycine residue of a protein. So that N-terminal glycine residue, well, that gets this myristic acid, which is, if I remember correctly, myristic acid is 14 carbons long. Yes, a 14 carbon long chain added to that N-terminal glycine. It is a glycine, but it's always that N-terminal glycine. So it's never going to be like glycine number 64. It's going to be glycine number, I believe it's 1 or 2. There are also thioether linked prenyl anchors. So we can have an isoprene unit, just like we saw on the previous slide, where these isoprene units, either farnesyl or geranyl groups, are added to a carboxy terminal cysteine residue that is then able to be inserted in the membrane and basically hooks in on the outside of the membrane and stays there. Now, our last group are these Gpi linked proteins, where we have what we saw on the previous slide, where we have our protein, then we have these sugars, we have our inositol group, and then we have a, this is our phosphatidylethanolamine, or our PE, or yeah, no, our phosphatidylcholine. Then we have all of these sugars, we have our inositol head group, we have the phosphate, that then this right here is our glycerophospholipid. So this is a glycosylphosphatidylinositol. So we have our sugars that bring in the glyco, then our phosphatidylinositol that gives our linkage to our membrane. So we have a phosphatidylinositol group that connects to a bunch of sugars that then are connected to the carboxy termini of our protein of interest. So these are the ways in which these are 4 unique ways in which a lipid linked protein can associate with a membrane. All right, well, I hope this is helpful, and have a good one.